research that supports restoration. We're really lucky to have Dr. Tanya Cheeke here. She's an assistant professor at Washington State University in the School of Biological Sciences. And her research is focused on how mycorrhizal fungi and the associated microbiome, below ground microbiome, can be tailored, and I love that word, can be tailored to improve productivity and resilience in both natural and agroecosystems. And of course, tonight she's going to talk to us about her research in prairie ecosystems and natural ecosystems. And with that, Tanya, I think I'll just leave it there and let you take over.
Okay, great, thank you. So I'm gonna try to share my screen here.
Sharing her screen, I just like to, I totally forgot to say that we will hold all of our questions until the end. And what I'd encourage folks to do is to put their questions in the chat box, and at the end of the presentation, I will bring in all of the participants so that you can have access to video and microphone, so you can follow up, and we'll go through the questions that we have in the chat box and have an opportunity to interact then. So apologies for forgetting to mention that ahead of time. So and back to you, Tanya.
Okay, thank you. All right, well, thank you so much for having me today. So as you mentioned, I'm Tanya Cheeke. I'm an assistant professor at Washington State University, and my lab is located on the Tri-Cities campus in Richland. And today I'll be telling you about some of the research my lab members and I have been doing, looking at the role of soil microbes in ecological restorations. And the goal of some of this work is to start to understand some of the mechanisms that can enable and promote native seedling establishment, with the overall goal of improving the biodiversity and function of disturbed or invaded ecosystems. So oftentimes when we think of the effects of disturbance, we often think of the effects on above ground plant communities. So you might imagine the impact of this clear cut on this forest ecosystem here, or tillage on this grass community, or effects of urbanization and road building. But it's also important to remember that the effects of disturbance that are felt above ground are also felt below ground in the soil ecosystem. The soil microbiome is made up of a complex group of organisms that are really important for both plant and soil health. And in my lab, we've studied each of these different types of organisms. There are symbiotic organisms like the arbuscular mycorrhizal fungi, ectomycorrhizal fungi, and nitrogen-fixing bacteria that form symbiotic relationships with plant roots and can help improve plant access to nutrients and water, for example. Also in the soil ecosystem are decomposers that break down organic material and recycle nutrients. So if you've ever been on a walk in the woods and have lifted up the duff layer, you might have noticed some of this white fuzzy stuff that's growing on the forest floor. And those are fungi that are breaking down organic material and recycling nutrients. There are also endophytes and pathogens that can have positive, neutral, or negative impacts on plant health. And then there are larger organisms like microarthropods, nematodes, and protozoa that are also important in nutrient cycling. And all of these different organisms have been shown to be negatively impacted by different types of disturbance. So this could be physical disturbance of the soil, so soil turning over. It could be the type of plant that's grown, whether it's a monocrop or a mixed plant community. And even the particular type of, the particular genotype of plant can have different impacts on soil organisms than others. And so in my lab, I run a soil microbial ecology lab at WSU. And today I'll be focusing on the arbuscular mycorrhizal fungi as part of this group. We've been focusing on the Arbuscular mycorrhizal fungi, or AMF, because these are one of the most ancient, they're thought to be one of the most ancient symbioses. They form symbiotic relationships with plant roots, and there's fossil evidence that they've been around for at least 450 million years, and they're thought to have helped plants originally establish on land. In this symbiotic relationship, the fungi form structures inside plant roots, so they live symbiotically with plant roots. The plants are photosynthesizing and they provide carbon to the fungi. And then the fungi here in orange create these thin hyphal threads that can go out into the soil ecosystem and access nutrients and water that are outside the root zone. And the arbuscular mycorrhizal fungi in particular are really good at getting phosphorus for the plant. And here's a picture of what these fungi look like under the microscope. So in our experiments, we'll harvest plants, a special stain that stains the fungi blue, and then we can look at them under the microscope to see how well the roots are colonized. So one of the things that's interesting about this symbiotic relationship is that the mycorrhizal fungi are completely dependent on a plant host. So they get all of their carbon from the plant, which makes them especially vulnerable to disturbance events that impact the plant. So for example, if this plant was harvested or eaten by an herbivore, these fungi would have to find a new host right away so that they can continue getting that carbon. They're also particularly vulnerable to disturbance because of these thin hyphal threads that they have in the soil, and they make these complex networks. So tillage can also break these up. Plants, on the other hand, have a whole range of responses to mycorrhizal fungi. So the fungi are completely dependent on the plant, on the plant host, but plants have a whole range of responses to mycorrhizal fungi. There are some, like this echinacea plant here, that are strongly dependent on mycorrhizal fungi and often won't grow in areas where the mycorrhizal communities have been disrupted. There are also plants like this Bromus inermis, or smooth brome here, that even when the roots are colonized with mycorrhizal fungi, they don't seem to show any benefit from that interaction. And then there are other plant species, like this Arabidopsis here, that have lost their association with mycorrhizal fungi over evolutionary time. So over 80 or 85% of plant species still associate with mycorrhizal fungi, but there are some groups like the Brassicaceae, so other plants in that family are like broccoli and cauliflower, have lost their association with mycorrhizal fungi over evolutionary time. And one thing that our lab and others have started to notice is that some of these plants that tend to be so strongly dependent on mycorrhizal fungi are some of these late successional native plants that are often only found in undisturbed environments. Whereas some of these other plants that seem to have no benefit or have lost their association with fungi over time tend to be some of these invasive and weedy species that can colonize disturbed areas quite easily. And that might be one of the reasons that they are able to, is because they know they don't depend on mycorrhizal fungi. So several years ago, Pringle et al. Did a meta-analysis where she looked at this question to see if native plant species had a stronger growth response to mycorrhizal fungi than introduced species. And so she looked at a bunch of different studies in the literature, and she found overall that the native plant species had a stronger growth response to mycorrhizal fungi than the introduced species. But other studies have shown that it also may have to do with plant successional stage or plant functional group. So it's really still relatively unclear what the best predictor is for plant response to fungi. Previous to my work at WSU, I was working at Indiana University, where I was doing prairie restoration within the tallgrass prairie system. And what I did was I had done a whole bunch of I had done some greenhouse experiments where I had taken a bunch of different plant species that were native and non-native to the tallgrass prairie system and looked at how they responded to inoculation with local or native mycorrhizal fungi. And then I took my data and I combined it with all the other studies that I could find in the literature who had also done something similar, so I could start to see if there were any patterns that emerged. And so what I found here on the x-axis are a bunch of the different tallgrass native plant species, and on the y-axis is plant growth response to mycorrhizal fungi. Above this one line is a positive growth response, and below is a negative growth response. And what I found was that these ones in purple, these are the late successional native plant species. And overall, they were much more strongly responsive to the mycorrhizal fungi than early successional native or non-native species. And in fact, many of these early colonizers, so the non-native and early successional native plant species, were actually even inhibited by mycorrhizal fungi. So they were smaller because the fungi can act as a carbon drain on the plant in some cases. And so what this did was this provided evidence that some of these late successional native plant species that have been more challenging to get reestablished in restorations may benefit from being inoculated with the local mycorrhizal fungi before they get reintroduced into a disturbed ecosystem. And in fact, several studies have been showing the importance of mycorrhizal inoculations in prairie restorations, particularly within the tallgrass prairies, where a lot of this work has been conducted. And even so much so that in this paper, Kojal and Beaver argue that mycorrhizal fungi are the missing link in grassland restoration. They can improve plant diversity and accelerate succession. And here's some data from the study that I've been doing in Kansas, where I have a tallgrass prairie restoration set up. And I inoculated seedlings ahead of time, either with whole prairie soil. So I went to a remnant prairie and collected soil that I inoculated with, or mycorrhizal fungi that were isolated from the prairie soil. And what I found was that the plants that were inoculated with the native organisms had higher survival rates than the uninoculated controls. And the ones that survived also grew better when they were inoculated with the local microbes. So here we have Echinacea pallida, little blue stem, which is a native grass, Lespedeza capitata, which is a legume, and Asclepias tuberosa, milkweed. And the colored bars are the inoculated plants. So they survived better and they grew taller when they were inoculated with these local soil organisms. So today I'll be telling you about some of the research that I've been doing now that I've moved to WSU. I moved here in the summer of August 2017. And in my lab, what we've been doing is studying plant microbe interactions within the context of changing environments. And so this is things like disturbance and biological invasions. So we study this from the individual to the ecosystem scale. One of the things that I'll be talking about today is our research looking at plant growth response to mycorrhizal fungi and mechanisms that could enable or inhibit the mycorrhizal symbiosis. And the reason that we're focusing on plant growth response to fungi is because we're interested in identifying plant species that might be more dependent on these mycorrhizal fungi than others. And that would indicate that those might benefit from some of these inoculations. In my lab, we also study the role of plant soil feedbacks in governing community structure. And then at the ecosystem level, we've been testing the efficacy of reintroducing native fungi to restore disturbed ecosystems. And so one of my experiments is in Kansas, and then I'll be setting up one here in the Palouse. And so the research that I'll be telling you about today is what my research program has been doing at WSU. And so I'll be focusing on the Palouse Prairie restoration projects that we've been working on. So over the past 2 years, we've been performing greenhouse studies where we've been testing how plants that are native and non-native to the Palouse Prairie respond to soil biota. We're also interested in understanding how biological invasions, or even do biological invasions, alter mycorrhizal function. There have been several studies in the literature that show that invasive species can have a negative impact on mycorrhizal fungi. And in the area that I've been working in, it's heavily invaded by cheekgrass. So we were interested in seeing if the cheekgrass invasion has a negative impact on the local mycorrhizal fungi. And then we're also interested in understanding what factors can influence variation in plant growth response to soil biota, including mycorrhizal fungi. And the goal of this work is to really start to determine which plants might benefit most from inoculations and start to understand the mechanisms that are governing variation in mycorrhizal response. We focus on the Palouse Prairie because it's one of the rarest ecosystems in the United States. And it's estimated that about less than 1% of the historic Palouse Prairie remains. The Palouse Prairie is over in eastern Washington, northwestern Idaho, and northeastern Oregon. And so here's a zoomed in map showing the historical vegetation. So in yellow, it was historically covered with native fescues and bunchgrasses. But since European settlement in the area, it's been converted almost entirely to agriculture. So the orange here is cropland, hay, and pasture, and a mix of exotic forbs and annual grasses. So it's really changed quite a bit. And the remnants that do remain are small and fragmented. So conservation efforts are probably not going to be enough to maintain the ecosystem services and habitat that these areas once provided. So active restoration efforts may be needed. So the native Palouse Prairie is comprised of cool season bunchgrasses like Idaho fescue and blue bunch wheatgrass, forbs, and shrubs. And so there's a lot of snowberry in this area. And so the Palouse Prairie has been converted almost entirely to agriculture, which has resulted in a loss of biodiversity and habitat for native wildlife and a loss of ecosystem services. One of the reasons this area has been so heavily converted to agriculture is because it has really naturally fertile soils. So the soil is high in phosphorus. And when you drive through eastern Washington, you see these beautiful rolling hills that are covered in wheat or dry beans. And so that's former Palouse Prairie. And because the remaining remnants are so small and fragmented, people have really been focusing on restoration efforts. But there are some challenges that come with some of these conservation efforts. So here's a project I was working on with some members of Phoenix Conservancy, where we were doing a native planting out near Smoot Hill. But these restorations restoration efforts can be impeded by legacies of disturbance. A lot of these restorations take place on former agricultural land where there have been decades of agricultural practices that can inhibit that can have a negative impact on mycorrhizal fungi. And they're also impeded by the invasion of non-native plants. So a lot of these restorations take place on areas that were former agricultural land that have had decades of tillage and monocultures and fertilizer applications, all of which can have negative impacts on soil organisms and mycorrhizal fungi in particular. There are also some pretty aggressive invasive plants in the area. So cheekgrass, medusa head, and wiregrass are some of the biggest invasive plant species that the people who are trying to do restorations are contending with. And so with all of these things, these could have potentially negative impacts on soil microbial communities. We know that agricultural practices can reduce mycorrhizal spore density and species richness. This is a study done by Ole et al. In 2005, where they looked at the spore density, so the number of spores per gram soil, of mycorrhizal fungi in grasslands, vineyards, and cornfields. And these different colored bars just represent different sampling depths. And what they found was that mycorrhizal spore density, as well as the number of different species, was much more reduced in the cornfields compared to the grasslands, which isn't surprising considering how sensitive these fungi can be to some of these management practices. Invasive plants there's also evidence that invasive plants can also affect mycorrhizal communities in soil. And so here are some of the main invasive plants in the area, the Bentonata bromistectorum cheekgrass and Medusa head. And so several studies have looked at the impacts of invasive species on mycorrhizal communities. And here's a study that was done by Vogelsang and Beaver in 2009, where they grew plants in soil that they had collected from an area that was dominated by non-native plants, and also grew plants in soil that was from a native plant area. And what they found was that colonization levels of the fungi in the roots were higher when they were grown in native soil compared to the non-native soil. And then they have some controls here. There's another study here showing invasive plants affect mycorrhizal fungi abundance and richness, as well as native plants performance in invaded soils. So there's a lot of evidence that invasive plants can have negative impacts on mycorrhizal communities, but there are also other studies showing either no impact or some of these invasive plants even can benefit from mycorrhizal fungi. So it's important to be able to test this in whatever ecosystem you're working in. We also know that soil nutrients can have a negative impact on plant microbe interactions, plant mycorrhizal interactions. And this is important because a lot of these restorations are done in former agricultural land where they've had years of fertilizer applications. So here is increasing soil nutrient supply, so increasing phosphorus in the soil, and this is mycorrhizal colonization in roots. So as the fertilizer levels in the soil increase, mycorrhizal colonization decreases. And this is because the plants are investing in these mycorrhizal fungi. They're investing carbon. And so when they're able to get all these nutrients for free in the soil, they're less likely to invest in this mycorrhizal network. So all of these different types of disturbances may give a competitive advantage to non-native plant species. And in the talk today, I'll be telling you about different plant successional stages. And the three successional stages I'll be talking about are the non-native plants, the early successional native plants, and the late successional native plants. And so non-native plants are fast growing. They colonize disturbed areas relatively easily. And so they may have a low dependence on mycorrhizal fungi. Maybe that's one of the reasons they're able to colonize these areas so quickly. Early successional native plants are somewhat fast growing. They can colonize disturbed and undisturbed areas relatively easily. So they may have an intermediate response to mycorrhizal fungi. And then these late successional native plant species tend to be slow growing. They thrive in undisturbed environments, and they may have a strong dependence on mycorrhizal fungi. And so with several different restorations that I've worked with and in talking with people, sometimes these restorations, people will add seed for 20, 30 years, and there are still some plant species that are really, really hard to get reestablished. And some of these late successional native plant species that are found really only in undisturbed environments. And oftentimes the diversity that's seen above ground is reflected in the diversity seen below ground as well. So here, if you have a mixture of plant species, you may also have a mixture of mycorrhizal species in the soil. Whereas if you have maybe a monoculture of corn, for example, and years of tillage and fertilizer applications and pesticides and all sorts of different things, that can have a negative impact on the mycorrhizal communities in the soil. And this can make restorations really challenging because if you're trying to get these type of plants to grow in this type of environment, it might be important to also think about restoring the below ground communities at the same time as above ground restoration efforts. But it's also important to understand which plant species might benefit the most from these soil inoculations. And that can be really important for informing restoration efforts. So determining which plant species might benefit from being inoculated ahead of time. So now I'll present some of the results from the greenhouse experiments that my students and I have been doing at WSU. And for the first research question, we've been focusing on how do plants native and non-native to the Palouse respond to inoculation with local soil biota? This work has been done on the WSU Tri-Cities campus, and here's a picture of some students and I setting up one of these experiments. What we've been doing is we've been performing these greenhouse experiments where we do these plant growth assays, where we test how different plant species, and we've been doing a mixture of native and non-native plants, to see how they respond to inoculation with local soil biota. And then we measure the plant growth response. And we can do this in two ways. One is a log response ratio, and one is a percent change in growth. So they both, basically, you look at how well they grew in inoculated pots versus uninoculated controls. And then we've also been doing some experiments looking at the functional potential of mycorrhizal fungi that have been collected from invaded and remnant soil. And the idea here is that at some point, what we'd like to do is create a database where we have, a list of native and non-native plant species in the area, and then have some information about how well they respond to mycorrhizal fungi. And eventually what we'd like to do is try to isolate and culture some of these mycorrhizal species that are local, that might be important in, they might offer drought resistance or access to soil nutrients, that we could culture in the greenhouse, and then people could use those either in their own restorations or in sustainable agroecosystems. So that's kind of the long-term goal. The work we've been doing out at Smoot Hill in Pullman, Washington. There is a biological reserve that's owned by WSU. It's about 800 acres, and it's near the WSU Pullman campus. And it's one of the largest intact remnants of native Palouse Prairie in the area. And it's been a great resource because students have access to it. There are several faculty research projects out there. There's a detailed plant list, and we have connections with local conservation groups. So it's been a really great resource for my students and I to use. And the research I'll be presenting today has been conducted almost entirely by undergraduates at Washington State University. And we also have received funding from the Murdoch Charitable Trust Partners in Science program. And this program sponsors a high school teacher to come and work in an active research lab for 2 years, where they can gain hands-on training in some sort of scientific process that then they can take back to their classroom and share with their students. And so here is Allison Winward, who is the high school teacher that I've been working with. And then here are some of the undergraduates and a graduate student who've been helping with this project. So in summer of 2018 and 2019, we went out to Smoot Hill in Pullman, or near Pullman, and we collected soil from an invaded area. So this area was invaded by cheatgrass. And then we also collected soil from a prairie remnant. And then we collected background soil also from the site that we used. We mixed 1-to-1 with sand and sterilized it so that we'd have a substrate for the plants to grow in. And then the only thing that changes in the pots is whether they're inoculated with the invaded soil or the remnant soil. And so we can see if there are differences in growth between invaded or remnant soil. The first year of the study, we used 8 different plant species. We had 5 native plant species and 3 non-native plant species. So the native plants were Venus penstemon, blanket flower, blue bunch wheatgrass, Idaho fescue, and monkey flower. And then our non-native plant species were cheekgrass, mullein, and white campion. And we grew them in these 3 treatments. And what we predicted is that the native plant species would have a stronger growth response to the microbes in the soil, in the local soil, than the non-native plants. And we also thought the native plants would have a stronger growth response to the soil organisms in the remnant soil compared to the invaded soil. To do these plant growth response assays, we have these little pots here. They're 1-liter pots. And in the bottom, we fill it with the sterile background sand soil mix. So it's autoclaved field soil, but just doesn't contain soil organisms. Then in the middle, we add 100 cc's or 100 milliliters of one of these different treatments. We top it off with sterile background sand soil mix, and then we put in a little seedling. And we replicate this for all the different plant species and the different soil treatments, and then we grow them in a greenhouse. So in this experiment, we grew them in the greenhouse for 4 months. And then what we do is we measure plant growth response to mycorrhizal fungi, or mycorrhizal responsiveness. And so this is typically the way the data are shown. So here we have the 0 line in the middle is no growth response. Here we have plants that have a positive growth response. And then here we have plants that actually grow worse when they're inoculated with mycorrhizal fungi compared to controls. And this again could be due to the plants investing carbon in the fungi and the fungi not returning nutrients in return. So here, these plants that have a positive growth response to mycorrhizal fungi, an example here would be this basil plant. If it grew twice as big with inoculation compared to controls, we'd say this has a positive mycorrhizal responsiveness. On the other hand, if it grows worse when it's inoculated, that would be a plant that has a negative growth response to soil biota. And several studies have been showing that a lot of the plants that have a negative growth to these soil biota tend to be some of these invasive or weedy plants. So one potential thing that we might be able to do is if we can identify plants that really benefit from the mycorrhizal inoculation, we could inoculate those ahead of time to improve the chance of those surviving and growing. And then also by restoring the mycorrhizal communities in the soil, we may be able to inhibit some of these invasive species. So plants were grown in the greenhouse for 4 months, and we collected data on plant height and leaf number. And at the end of the experiment, the plants were harvested and we collected data on biomass, on the above ground and below ground biomass. And then we used that to calculate mycorrhizal responsiveness. And we also collected root samples so that we could look at the level of mycorrhizal colonization in the roots. What we had predicted for these first 8 plant species, we had predicted that the invasive species or the non-native species would have a negative or neutral growth response to mycorrhizal fungi. And we had predicted that these native plant species would have a positive growth response to the mycorrhizal fungi in the soil inoculum, and that there would probably be some variation in response on both sides. And the invasive species are going to be in orange, and the native ones are in blue for the next few slides. And what we found was when we looked at all the data, we found that the growth response to the local soil biota was species specific. So we had predicted that the native plants would have a positive growth response and the invasive plants would have a negative growth response, but it was actually quite a bit more mixed than that. Supporting our predictions, cheekgrass and common mullein were inhibited by mycorrhizal fungi, so they grew worse in the inoculated pots compared to the controls. Venus penstemon is a native plant, and that benefited from the inoculation, but so did one of these invasive or non-native plant species, Silene latifolia. And this also happens. There are some invasive plant species that do take advantage of some of the mycorrhizal fungi in the soil. But then some of these other plants had a sort of a neutral response. If this little tail here goes over the dotted line, it means there was a lot of variation. So the next thing we did was we were interested in understanding whether invasion by cheekgrass could alter the mycorrhizal inoculation potential. So we collected that soil from the cheekgrass invaded area and the remnant area, and we wanted to see if that had any different effect on plant growth. What we did was we conducted, it's called a mycorrhizal inoculation potential assay, and it's a really quick greenhouse experiment where you can test the function of soil biota. So what we did was we grew up a bunch of these little corn plants, and we inoculated them either with soil that came from the remnant prairie or soil that came from the cheekgrass invaded area. And then we looked at roots to see how well they were colonized. And this gets at the function of the mycorrhizal fungi, because oftentimes the more colonized the roots are, the more nutrients the plant can get. And we predicted that the remnant soil would have a higher mycorrhizal inoculation potential than the invaded soil. But to our surprise, we found that invasion by cheatgrass did not alter the mycorrhizal inoculation potential in this experiment. So here we have the invaded soil and the remnant soil, and this is percent colonization of roots when the plants were grown in each of these two treatments. And so this was surprising to us because almost every study that I've read has shown that cheatgrass can have a negative impact on the mycorrhizal communities in the soil. But one of the things we don't know is we don't know how long ago this area was invaded. And we've only collected samples so far from those two areas, or that one area, those two samples. So I have two new PhD students who joined the lab this year who are interested in collecting samples in more areas throughout the Palouse Prairie bioregion to see really what's going on here with the cheatgrass, if this is really consistent or whether we'll see variation in results if they collect more samples. So in the first year, we found that plant growth responses to inoculation were plant species specific, and we really didn't have enough different plant species to be able to start making predictions yet. We also found, that, to our surprise, that the Palouse Prairie, that area that was invaded by cheatgrass, actually maintained the same mycorrhizal colonization potential as the nearby remnant prairie soil. But we don't know how long ago it was invaded or how consistent these results will be. So this is one of the next things we're going to be testing. And then we don't really have enough data yet to make predictions about plant growth response based on native plant status or plant successional stage. So for the second year, we added 17 more plant species to our experiments. And the ones that I'm showing you here today are the ones that, so we got seeds from different sources and we tried to grow up a bunch of different species, but the ones that I'm presenting today are the ones that grew up in high enough numbers that we could use in our experiment. And so the seven native plant species that we used in the second experiment were Western mountain aster, prairie smoke, Wyeth buckwheat. We looked at Idaho fescue again, sandbird bluegrass, showy milkweed, and sticky purple geranium. And then we had a whole bunch of different non-native plant species, that some of them that had been recommended by local restoration practitioners as being problematic in the area. So like wiregrass and rat-tail fescue and medusa head. So we did the same type of experiment where we grew the plants with and without the local soil biota in the greenhouse. And then we collected growth data to see how well they responded to each of the treatments. And what we found again was that these invasive plant species tended to be inhibited by the mycorrhizal inoculum. So they grew worse in the pots that were inoculated with this local prairie soil. And then we had two native plant species that seemed to benefit quite a bit from the inoculation. So this was Asclepias speciosa, or showy milkweed, and Wyeth buckwheat. But then again, there are several in the middle here that don't seem to show a strong responsiveness either way. And so in each individual experiment, we see some variation in results, but so far we have collected data on the mycorrhizal responsiveness of 24 different plant species that are native and non-native to the Palouse. And what we have been finding is that pretty consistently, the mycorrhizal inoculations inhibit some of these invasive plant species, but also that hardly any of the invasive plant species benefit. We've only found one so far that has benefited from the inoculation. Whereas some of these native plant species have benefited, or they do grow better. Let's see. Yeah, and so in the future, what we would like to do is really focus the next set of experiments on some of these more late successional, rare, endangered, imperiled Palouse Prairie species. And we have tried some of these in the previous studies that I mentioned, but we had a hard time getting enough of them to grow in time for the experiment. And so this next round, what we're going to do is just focus on this group of plants or plants that are that are similar. So things like Palouse thistle, Jessica's aster, Spalding's catchfly, to see how they respond to the mycorrhizal fungi in the soil. Because these are the ones that I would expect that would have the strongest growth response. So what is driving variation in these results? We have all these plant species specific responses, but it would be great to collect enough data that we could start to make predictions about which plant species might benefit the most from inoculations. Is it native plants? Is it late successional native plants? Is it a particular functional group? It's also possible that variation in nutrient requirements are driving some of this variation. And then also it's possible that the source of mycorrhizal fungi might be important for these inoculations. So to test some of this, we looked at these different successional stages. So we have the non-native plants, the early successional native plants, and the late successional native plants. And we started to look to see if there was any pattern in their response based on the data we have so far. And in the tallgrass prairie system that I had been working with previously, there was a very strong gradient where the late successional native plant species were much more responsive to the mycorrhizal fungi than early successional or non-native. But it hasn't been looked at in the Palouse. There has been some work, this was done in 2007, where some folks looked at mycorrhizal responsiveness of plant species in Rocky Mountain National Park in sagebrush steppe vegetation, and that they found that some of these later successional plant species had a positive growth response to the mycorrhizal fungi, which would follow our predictions, and that some of these early successional native and non-native plants were inhibited. And they also found that Bromus tectorum cheatgrass was inhibited by the mycorrhizal fungi. So what we did with our data is we assigned these coefficient of conservatism scores to each of the different plant species. And these CC scores can be used as an indicator of plant successional stage. And there's this website that you can go to. It's the Universal Floristic Quality Assessment website. And what you can do is enter in the name of your plant species, and then it gives you a number, a CC score. And these numbers range from 0 to 10. And these CC scores are indicators of a plant species' sensitivity to anthropogenic disturbance. So plants that have a CC score of 0 are things like cheekgrass that can grow pretty much anywhere. They are not sensitive to anthropogenic disturbance at all. Whereas the higher the number, the more sensitive they become. So something with a CC score of 10 would likely only be found in undisturbed remnant prairies compared to something with a lower CC score that could grow more easily in disturbed environments. So we looked up the CC scores for each of our plant species and did just a little preliminary analysis because we do still need a lot more data and to test a lot more species. But we do have some of these, we had enough of Jessica's aster and Spalding's catchfly to use for this analysis, but not for the full factorial greenhouse experiment. So we do have representation from each of these different different successional stages. And what we had predicted was that as the CC score, as the coefficient of conservatism score increases, so the plants over at this end are the ones that are more sensitive to disturbance, and these are less sensitive to disturbance. We plotted the CC score against the response to mycorrhizal fungi. So here's a greater response, and this is a lower response. And what we would predict is that if these were related, we would have this kind of a straight diagonal line here with an increase. And we see some evidence of that here. This Silene latifolia doesn't fit the pattern that we were expecting, but that happens. There are plants, there are non-native plants that can benefit from mycorrhizal fungi. So what we'd like to do next is fill in this area more with some of these more later successional, rare, or endangered plant species to see if that follows the pattern that we are expecting. We also did an experiment to try to get at another mechanism of looking at how these different plants respond to added nutrients. On the x-axis here, this is increasing nutrients that were added. And then this is increasing response to the local soil biota. And what we found are that the plants that responded most to the added nutrients also responded most to the added organisms. And so what this suggests is that these plants may have higher nutrient demands that they're able to meet either through fertilization or by forming associations with these organisms that can help them get more nutrients. So what we found in these first 2 years of studies is that the plants native and non-native to the Palouse vary in their response to soil biota, which may be related to nutrient demands. We found that soil biota function isn't always altered in invaded soils, but this is contrary to a lot of what is shown in the literature. So we need to do more research and test more sites to see if this result is consistent. We also found there was a trend of increasing mycorrhizal responsiveness of native plants with increasing CC score, but more data are needed, especially up at that higher end with the CC scores of like 7 to 10, which will be the focus of our next set of experiments. So I hope in this seminar today, I've convinced you that mycorrhizal fungi are important for the growth of some plant species, even in high nutrient soils. So the soils in the Palouse are high in phosphorus already, and the plants still, some of them still benefit from this inoculation. And these growth responses may be related to an increased access to nutrients. We found that the soil biota in at least this one degraded prairie site might remain functional. So this is important because if you're interested in using soil inoculations in restorations, it's really important to test the function of the soil biota in that particular site. I mean, if the soil biota is functioning as usual, then it may not need to be inoculated. But having information about which plant species benefit the most, and also some information about the function of the resident organisms in the site, will help to determine if inoculation is needed for a successful restoration. Because inoculations can be expensive and they can take a lot of time. And so there might not be a need to use it in a particular situation, whereas in another situation, they could be really beneficial. We also showed that plant species vary in their response to soil biota. So considering these different factors, plant species responsiveness and soil biota function, we really hope can help managers plan plant mixes and restoration strategies if people do use inoculations so that they can use them most efficiently. So with that, I'd like to thank all the people who've helped in this project. As I mentioned, most of this research was conducted by undergraduates at Washington State University. And here are a list of some of the names of the people who've come through the lab. There have been, I think, about 25 undergraduates who've received training in the lab so far. And many of them have gone on to get jobs at PNNL or gone on to graduate school. We've also received help from WSU faculty and staff for the greenhouse experiments. And we've been working with some folks in Phoenix Conservancy who've helped us with the project. And a lot of the seeds were provided by Thorn Creek Native Seeds or were collected locally. And we have gotten some helpful suggestions and advice from local conservation experts, JC Jensen, Brenda Earnhardt, and Trish Heeken. So with that, I thank you for your attention, and I would be happy to take any questions.
So there are some questions in the chat box there, Tanya. If you want to start with those. I think Nikki had a question about whether AM fungi inhibit or prevent invasion by non-native species.
Yeah, that's a good question. So we've found in several of these greenhouse studies, both in the Palouse area and also in the tallgrass prairies, that the AM fungi can inhibit some of these invasive plant species for sure. And I think that more intact remnant prairies that are, have less disturbance will be more resilient or resistant to invasion. So whether or not we can, the idea is that I would really like to test is whether we can restore mycorrhizal communities in disturbed ecosystems in such a way that it will help these native plants survive better, grow better, and compete better with these non-native species. And that would help hopefully keep those out. But that's one of the experiments that we're hoping to do in the field. Okay, so here is mycorrhizal inoculation potential species specific. So the arbuscular mycorrhizal fungi tend to be generalists, and this is because they require a plant host for their nutrition and reproduction, so they can't be too picky. Ectomycorrhizal fungi are a different group that form associations with trees and shrubs, and those tend to be more species specific. But the arbuscular mycorrhizal fungi are considered to be generalists, so they tend to colonize whatever plant they can get to. Okay, is there a known trajectory timeline of recovery for AM fungi and below ground microbiome without intervention? So this is a tough question because the work that I was doing in the Midwest, the restoration that I was doing was fairly close by to a local remnant prairie, but these fungi are dispersal limited. They live underground, they're microscopic, and they don't travel very fast or very far. And so even when there is a local reference site nearby, you can also have sites where the fungi just aren't getting there. And so one of the things what we're trying to do is do these inoculations where we take a small amount of soil from a remnant prairie and either inoculate with that soil, or we isolate the fungi and culture them and then use those to reintroduce into a disturbed system. And because some of these restorations have been going, at least in the Midwest, sometimes for decades, and there are still some plant species that just won't take. But these tend to be the ones that are so dependent on mycorrhizal fungi. So inoculating them or adding fungi back into the system might help them establish better. Why are there circular areas where cheatgrass does not grow while it is prominent nearby? I have no idea. That would be a great question. We could collect soil samples and look and see if it has anything to do with soil organisms. That's a great observation. Let's see, do you think soil inoculation would help with restoration of other landscapes besides prairies? Yes, so another part of my research program is I'm developing a grant proposal to test this in forest ecosystems after wildfires because the fungal communities can be negatively impacted by forest fires. And so one of the things that I'm interested in is, how these different mycorrhizal communities are impacted by fire and whether soil inoculations can help increase the recovery or improve the recovery time. And people are also using these. So I have another component of my research program where we're using mycorrhizal inoculations for wine grapes because that's a big industry around here. And it's a way to use these organisms as a potential biofertilizer. And then once you add these organisms to the soil, as long as you don't kill them or they don't get out competed, theoretically they should self-propagate. And so it would be a way for people who are growing vineyards to maybe reduce fertilizer costs and be able to use these organisms. How many times would you have to inoculate and for how long a time frame before these mycorrhizal fungi establish without need for intervention? That's another thing that I've been testing in my restoration site in Kansas. What I found is that it seems like the fungi are moving about maybe a half a meter a year. So I put in a row of inoculated seedlings and I've been measuring using different plants, like how far these fungi are traveling. They're also going to be competing though with whatever resident fungi are there. So that's a really open question that I think people would be very interested in learning more about. And so the experiment that I'm doing in Kansas is trying to get at some of that. What is the moisture level of the remnant soil? It seems that it would be dry and therefore inhibit microbial activity. Okay, that's another great question, and something else that my new grad students will be looking at, because coming from the tallgrass prairie system, it's hot, it's humid, there's a ton of rain in the summer. So it's a totally different system than these more semi-arid ecosystems. And the greenhouse experiments that we did, we have these automatic waterers. So the plants are like well watered and they're grown in ideal conditions or greenhouse conditions. But one of the things that would be interesting is to do these experiments at different moisture levels, because the fungi, maybe in these high nutrient soils, maybe they're, maybe they're helping with some nutrient uptake, but maybe the real thing is maybe they're helping the plants get moisture, for example, helping them get water. So that would be something that I'd like to do in the greenhouse experiments as well. How much variation is there in the specific microbiome organisms from one location to another? Yes. For instance, are the organisms in two different Palouse Prairie patches the same, whether they're 20 or 200 miles apart? This is a great question, and one that a lot of people are working on right now. And that's what we'll be doing in the Palouse Prairie bioregion with my new graduate students, is we're going to be collecting soil samples and then sequencing to see, what are the mycorrhizal communities in invaded areas compared to the remnants, because soil is really patchy, and it also varies by depth. So depending on how deep you sample, there are some organisms that are going to colonize closer to the top and some that are deeper. So it's a really, really great question, and so one that we'll hopefully have some data on in a few years.
There's also a couple of questions in the question and answer box from one of them is, how are you determining what native plants to focus on? Are you asking whether you're coordinating with local tribes? That's the first one.
Mm-hmm. Okay, so that's something, so I moved here in August 2017, and I started making connections with different people in the area to try to get an idea of what plant species of interest people would suggest that we start focusing on. And so I had met with some people over in Eastern Washington. JC Jensen was one of them. So some people who are interested in conservation, who have been working on conservation for quite a while. And I had a spreadsheet of all the different plant species that we could start to look at. And they gave me some ideas of which ones would be nice to focus on, to start with some of the most invasive species and also some of the ones that are the native species of interest. Again, a lot of the seeds, or some of the seeds, didn't grow in enough quantity for us to be able to use in the experiments. And so some of those plant species that we had hoped to focus on, we weren't able to get enough seeds or enough plants to do that with. We have not coordinated with any tribes so far, but that's part of the plan as we expand, as we start to develop these research plans. I had met with several people at the Society for Ecological Restoration meeting a couple of years ago. And so I have some connections. And now that we've collected some preliminary data, that's one of our next steps, is to start talking to other stakeholders, see what other people are doing, where our research can fit into that. And then I see, do you have any plans to do any mycorrhizal community analysis to assess differences between those that inhabit invasive versus natives? We could do that. Right now we're planning on sequencing the soils. We could also sequence the roots, the roots of the plant. So that's another thing that we could do too, to see if we could grow invasive and native plants in invaded or non-invaded soil, and then look to see if they have differences in the communities that are colonizing their roots. So that's a good suggestion.
There's another one in the chat box from Pauline Schaefer.
Okay.
I was just going to let folks know that I've made everybody panelists, so you all have access to video and microphone. So if you do have follow-up questions that you haven't put in the chat box, or you can raise your hand and we can try and recognize you that way. Otherwise, I think we'll be talking over the top of one another. So.
Okay, so Pauline asks, in your intro, you mentioned that some plants have lost their association with fungi over evolutionary time. How do we know that they ever had an association? So the best evidence for this is that there are fossil records of plants with mycorrhizal fungi, or what look like mycorrhizal fungi, in their root systems from 450 million years ago. And the kind of current hypothesis is that that's what helped plants first establish on land. So plants are thought to have originally been aquatic, and then when they moved on to land, having these associations with mycorrhizal fungi, the fungi could act as sort of like pseudo roots and help the plants get established on land. So I guess we don't know for sure that they ever had an association, but most plants still do. So 85% or more of plant species still have these associations. And there are just certain groups that have, people think they've lost that association over time. So they must have developed other strategies for getting nutrients and water, for example, that don't rely on fungi.
Wow.
Any other questions? These are all great questions.
Lots of great information. So, did you get to both questions in the question and answer? I think I was, okay, sorry, I didn't hear the second one because I was busy promoting people to panelists. So.
Oh yeah, no problem. Yeah, that was about the fungi and roots versus soil, and that's something that we hope to be able to test at some point too.
Okay.
All right, well, those are some great questions. I don't know if there are any more. It's already about 8:15, so I don't know if some people need to leave, but thank you for your attention and for having me. And if you have other questions, you're welcome to reach out via email or my Twitter address or anything like that. Well,
I would just really like to thank you for giving this great presentation. So hear from people in the chat box is so interesting, and we all really appreciate it. I learned a lot and really enjoyed hearing about your research. So.
Yeah, and you know what I would like to say, since I am relatively new to the area, and also my 2 new PhD students just moved here from out of the area too, you know, we have a lot that we could learn from you and your group. And so I really hope to start building more connections, because I'd love to get your ideas about what plant species should we test next? What would be of interest? Or what have you noticed in your restorations? Or are there certain plant species that are harder to establish than others? Like, that's the type of information that would be really helpful. And then, you know, working with these undergraduates at WSU, we can do these experiments in the greenhouse like over a summer.
Yeah, it would be really interesting to have some further discussions or be in the field together for sure.
Mm-hmm.
Yes, I keep seeing circular areas where cheekgrasses died. There's lots of cheekgrass right next to it, and these have been recognized. And there's cheekgrass die-off, and it's a bacteria. I'm not sure. Maybe it needs to be looked at again from a different perspective.
Yeah, that would be super interesting, because yeah, if you can find a biological control for that, that would be great.
Well, yeah, I could probably collect some soil samples for you from the circular areas and the nearby cheekgrass areas and give it to you.
Yeah, that would be interesting to look at. Yeah, and I know you gave me some sandberg bluegrass before, yeah. And I don't know if you saw it or not, but we included it in one of our studies, and it was not responsive. So it was one of the native plant species that, at least in our experiment, did not have a strong growth response to fungi. But it may be more of an earlier successional native, and so maybe it doesn't have such a strong dependence on it as others.
Well, I'm noticing this too in my field experiments. There's just no effect.
Mm-hmm. Yeah, and so that's why, that's why what we're hoping to do is like do a bunch of these experiments and then make these data publicly available, so that when people are working on restorations, you don't waste your time and money on inoculations that won't benefit that particular plant. And so it can really maximize the efficiency, I think, of using some of these ideas.
Yeah. There's all sorts of trouble getting native plants established in restoration. We don't know enough at all.
Yeah, well, send me your lists. Tell me what you want us to test next, because I think we're done looking at the invasives. They pretty clearly don't, for the most part, don't benefit from the fungi. So what we're trying to identify are native plant species that really could. So that's our next step.